Our last speaker is Jeff Riffell. Jeff is here at the University of Washington. He studies insects like moths and mosquitoes. Today he's going to be speaking about moths, which he likes because they are fuzzy and they are the butterflies of the night.
Yes, they are. Thank you. That was wonderful. Yeah, I work with mosquitoes as well, and yeah, moths are like this kind of fuzzy, very cute organism. Okay, so I'm going to talk about some of our work on olfaction in the Anthropocene. Nonetheless, I'm at the University of Washington, and welcome to Seattle. Hopefully you can actually visit some of these locations around town. I know many of us at the Allen and Kavli and et cetera, many of you have already been here. There's a lot of great things to go and see. Nonetheless, I'd like to introduce you to other side of Seattle. Seattle has a lot of celebrities like Kenny G, Dave Matthews, etc. Another celebrity is Good Space Guy. Okay, and so this guy was on the ballot of every ballot up into like 2022, and you see him every once in a while. The thing that's frightening is that you get about 4 to about 48% of the ballot people will vote for him. So he keeps the Seattleites kind of on their toes in terms of who we're hiring for our governor or senator or congressman. The other interesting thing about Seattle, it's a local community of T-Rexes. Every year, T-Rexes will kind of join as a collective behavior and go and perform a race in town, and you actually see them in town. So it's a very interesting habitat for them. The other thing is if you don't want to go to the Troll or the Space Needle or something, there's a really interesting gas station kind of by my house. It's a Wallingford gas station, and they have wonderful signs. Ban pre-shredded cheese, make America great again. Ants are healthy because they have little antibodies. Oh, that's great. And then all those who believe in psychokinesis, raise my hand. So this is, so this is Seattle for you guys. I hope you can enjoy this town. It's wonderful. Okay, so I'm going to talk to you today about some of the work that we have going on in the lab. And it's really, the focus is how do pollinators find their patches of flowers. And many of these pollinators, we don't realize it, but they're nocturnal. They can fly up to kilometers. This moth, the Manduca sexta, has one of the most sensitive olfactory systems in the world. And it will actually navigate to patches of flowers over kilometer scales. It actually flies up to 80 kilometers in the evening. It'll visit the flower. It'll pollinate it. They're really important pollinators. Pollinators are really critical for the plant community. Okay, they are shaping the community that we have around us, but we don't really know very much about many of the pollinators that are flying around at night. Nonetheless, they have really critical roles in our food security. Up to about 75% of the food that when you go into the produce in the grocery store, you actually is probably mediated by pollinators. And so without those pollinators, we don't really have that much great food. Nonetheless, we saw some noise pollution, there's light pollution. Olfactory pollution is also really shaping kind of the sensory environment for many of these organisms. Olfaction in general is the least or less studied sensory modality, so we don't really understand how these sensory pollutants are really shaping the animal behaviors. Nonetheless, they are shaping us. Many of the disorders that we have, say like asthma, cancer, neurological disorders, heart and lung disease, etc., are through these particulates that are out in the environment that are through these anthropogenic pollutants that we're releasing into the environment itself. So over the last several years, we've been really interested in understanding how these pollutants, these airborne pollutants, are actually shaping plant-pollinator interactions. There has been increasing work in these domains. Colleague Robbie Gerling has found that diesel exhaust really can impact pollinator behaviors in some plants. Nonetheless, diesel exhaust is all sorts of different compounds in the environment. There's all sorts of things in there. We don't really understand very much about what is actually shaping and modifying the plant behavior. Ozone is another pollutant, but it's a daytime pollutant. It's also natural. and nighttime has different atmospheric processes, which I'll get into. If you actually look in the environment, some of these pollutants are actually natural, like ozone. It fluctuates. It's actually produced by atmospheric processes, and different areas will have different amounts of pollutants. If you actually go to Yosemite or the North Cascades or out in the forest, you'll actually see this haze in the kind of daytime. And that haze is through ozone-mediated particulates that are reacting with some of the volatiles that these plants are producing. But at night, it's a total different process. In this case, these nitrate radicals are the dominant pollutants in the environment, and they are shaped, they actually react, photo-oxidize, and they won't be present in the daytime, but they're really abundant at night, and they're really having a really strong effect on these kind of atmospheric particulates. These are the NO3, or these nitrate radicals, are the things that are actually causing the smog in these urban environments. There's the free radical that's causing this particulates that's causing us to have asthma. And so they're really important for control, I guess, environmental regulations. We're an olfaction lab. We also do a lot of chemistry. We're really interested in understanding how do animals pick out an odor source from a very complex olfactory environment. If you actually look at this flower, it's producing like sub-micrograms per second per meter squared. But if you actually look at the volatile kind of emissions of some of the neighboring environment, it's actually 5 orders of magnitude higher levels of emission. And what's kind of fascinating about this is that these kind of emissions can actually be really similar in both the chemical profile that they're producing, the compounds. And so how do pollinators actually recognize this flower amongst this kind of bouquet or melange of competing odor sources? And then finally, how are these anthropogenic pollutants actually shaping the ability of these animals to actually recognize these odors? So these are some of the questions that we're actually exploring in the lab. We use a variety of different kind of approaches. We do a lot of chemistry. We try to understand how the brain is actually representing these different odors under these different regimes, both pollutants. And then we look at behavioral impacts both in the laboratory as well as in the field. And I think the way that we are trying to understand this and kind of link these processes is actually using a variety of different kind of 3D thermokinetic models to actually explore how different global areas are actually being impacted by these processes, and also using a variety of different ML tools. And so some of the questions we're asking is, like, how do these anthropogenic pollutants, how are they impacting the odor, okay? One idea is that they are actually just impacting and just kind of degrading all the odor compounds in the air around us. Another idea is that they are just kind of selectively removing certain key features of the bouquet, if you will. And then the other idea that no one is really certain about is that there's evidence that they're actually producing new compounds that in turn the animals can detect and might be modifying the behavior. But we didn't know at this point which of these features were actually being involved. So to do this, what we used is we did a lot of headspace volatile analysis using a proton transfer reaction time-of-flight mass spectrometer or a chemical ionization mass spectrometer. We could actually release these volatiles from, say, a flower into a flow tube that we could actually control the reaction with a known pollutants, say like ozone or nitrate radicals. And then we can look in real time, measure how these volatiles are actually being kind of degraded, and look at also the products that are arising from this. And so what was kind of interesting about this is that if you actually put in this complex mixture of different flower volatiles, if you smell a flower like a rose, you're smelling 300 to 1,000 compounds at the same time, but you can actually recognize it as a rose. We can measure this with the mass spec. What we found is that certain compounds within this complex bouquet would react pretty readily. By contrast, other compounds like this aldoxime was completely robust to any sort of kind of chemical pollutant that we put into there. It wasn't reacting, in other words. By contrast, some of these terpenes like beta pinene, beta-ocimene, et cetera, it would react to, say, ozone, but if you give it some nitrate radicals, all of a sudden it would go down to close to zero, up to 80% of it would be lost. So what's happening in this environment through nitrate radicals is actually just kind of removing certain features of the volatiles within the bouquet. So one of the things we wanted to do is like, how do these anthropogenic pollutants, like these nitrate radicals, how do they react with the odor, and how does it modify the animals? And so we did these experiments both in the laboratory but in the field. This is actually, I didn't do this. This is Jeremy Chan, who is a graduate student in the department, in the lab. And what he's doing is this face experiment, where he's actually releasing nitrate radicals around these patches of flowers and then seeing how that modifies the pollinators that are visiting these flowers. He did this with real flowers and flower scents. He did this with, he was releasing degraded odor from these kind of artificial flowers. And then you just have artificial flowers that just looked precisely like a real flower, but it was just not emitting anything. It's like a visual control. What he found and what we found is that if you have a flower odor and kind of a visual cue, the pollinators, especially these moths, the Manduca sexta moth, would visit it really readily. By contrast, if you oxidize the flower scent from an actual real flower or, say, an artificial flower scent, all of a sudden, kind of the moths would no longer visit these flowers in their natural habitat. And this was equal to this kind of no odor control, which is just this visual display of the flower itself. Kind of this artificial visual display. And so what this actually translated into in terms of kind of pollinator visits or moth visits to these moth-pollinated flowers, actually decreased pollinator plant fitness in terms of their kind of fruit and seed set by up to 25%. Okay, so it really decreased the plant's fitness by having these anthropogenic pollutants in the environment. So how is it actually impacting their representation? How is the olfactory system impacted? And so one of the things is that because these nitrate radicals, these anthropogenic pollutants, are actually removing certain compounds, it's actually altering the excitation inhibition in the olfactory system or the antennal lobe. You can think of this as that the moths are actually encoding this floral kind of complex blend as a precise kind of ratios. Ratios are really critical, and in order to actually kind of encode these certain features like beta-pinene and kind of ocimene, you have to have a precise inhibitory connections through these GABAergic inhibitions. If you modify this proportions, then all of a sudden the representation is really modified. And you can see this through these intracellular recordings. If you just increase the drive or the concentration of this yellow ORN to PN, then all of a sudden you get a much different representation than if the excitatory balance is maintained through the precise ratios that the flower is producing. We could also do this through kind of multi-channel recordings. We can record from many neurons simultaneously. We could actually release kind of these nitrate-modified nitrate radical kind of odors, just a real flower odor, et cetera. What we find is that if you maintain and kind of keep the flower odor the same, then the neurons can actually encode the temporal dynamics of the odor stimulus very precisely. By contrast, if you give the moth these discrete pulses or a background of NO3 or a modified flower scent, all of a sudden this balance of excitation and inhibition in the system causes the representation of the flower odor to be totally modified, and it becomes unable to really accurately encode the temporal dynamics of the odor. So in this case, the anthropogenic pollutants are really shaping the ability of the moth antennal lobe to kind of encode the representation of the flower scent. So one of the things, so this is great. So this moth goes from Central America up to North America. These compounds that that are really sensitive to anthropogenic pollutants are found in all these different flowers. So we actually took this system and say, okay, what is it like for other regions of the globe? We use this open access, this open model that you can actually play around with, Geoschem. It's a fantastic atmospheric model, science model, where you can actually, if you know the regional levels of these nitrate radicals, as well as the concentrations of these compounds, then you can see how quickly they're going to be reacting or being modified in these different regions of the globe, okay? And so what we did is we took this, and because we knew the kind of concentration that the moths could no longer recognize the flower, and because certain nitrate radicals like NO3 is going to react with the flower scent, with this model, it's fantastic because you incorporate meteorological aspects into it, like wind speed. Because the nitrate radicals are going to be reacting with the flower scent, with the wind speed, you can then predict how far the odor plume is going to be transmitted for the moth to recognize it. And what we found is that if you live in Southern California, sorry, San Diego, the moths can no longer really recognize the flowers. If you're in Western Europe, you're also, these pollinators are also kind of screwed. Other regions of the globe are actually fine. So different areas are going to be really differentially impacted. The great thing about these models that I think we really need as this part of this community, what these models can do is that we can go back in time, and then we can go actually forward in time and see how is it impacting these behaviors, these pollinators, and these plant communities. What we find is that what's really fascinating is that certain areas, we can look, certain areas are actually impacted more, say Southern California, Spain, et cetera. There's also, say, Egypt. What's really fascinating about going back in time and comparing, though, is that these areas are relative to pre-industrial, like Southern California was really impacted, but if you go up to Egypt, Egypt was historically already had these really high levels of these naturally produced ozone or nitrate radicals. And so certain areas of the globe had naturally high levels of these pollutants, where other areas are due to anthropogenic inputs alone. So this raises the question is, how have these different areas, these animals and these other different areas, how have they evolved? And so this is one area that we're really excited to explore and see how these different regional areas are, how animals have actually been impacted by natural versus anthropogenic pollutants. So I think this kind of framework and then trying to actually use climate models can be a really effective way to lead to new questions that we should be asking. So with that, I'll just really thank members of my lab, especially Jeremy Chan and Cleo Pimienta, who's actually here somewhere. Joel Thornton is a wonderful collaborator, and I'll take any questions. Thanks.
Sorry for the Zoom people. Any questions?
It's time to
Yep,
eat.
right here.
Yeah, okay.
Yeah, wonderful. Have you looked at plume tracking in the behavior and see if that's affected? Because that's usually turbulent air, right? And so the concentrations go up and down naturally in there.
Yeah.
And I don't know if the different compounds change in the turbulent.
No, it's like so this is actually, you can actually quantify or determine through distance how far, at what point in distance will it actually, the moth be able to not recognize it. And usually, so like in Los Angeles, it's now about 5 to 10 to 12 meters, where traditionally it was like a kilometer scale. You know, so the moth, if it gets close enough, it will be able to recognize the flower scent because it doesn't have enough time. And at which point it's using what's called a cast and surge model to actually kind of locate these patches of odors. I should say that one of these weaknesses of these atmospheric models is that the grid size is huge. You know, it's anywhere between 1 kilometer to 200 kilometers, okay? And so what we really need is a model to actually go from kilometer or sub-kilometer scale to something that's called a large eddy simulation model, which is like meter scale, to even sub-meter scales. And so this is a need for the climate scientist to meet with the behavioral ecologist / neuroscientist to try to kind of get at these models to make more predictive information for the behavior that we need. That's my plug.
And then, yeah, Alok.
Yeah, thanks for that talk. I was wondering, since these nitrate radicals seem to be circadian, what do you think is happening to daytime pollinators? Are they being affected by any radicals and volatiles, or do they just not
Yeah,
are they less affected?
You know, that's a great question. So what's going on for the daytime? That's something that we want to pursue, and that's something kind of I'm trying to get colleagues here to work with us on, is that daytime pollinators are subject to completely different atmospheric processes. They are subject due to ozone and hydroxy radicals, and they will in turn have their own kind of timescales and modify other kind of chemicals in the odors. So it's interesting. Yeah, daytime is totally different than nighttime.
Okay, but have you looked at whether the distance that they can detect is still reduced compared to what you would expect from?
Yeah, I mean, what we found is that ozone, which is the daytime free radical, is not as does not have the impact that these nitrate radicals do at night.
Cool, thanks.
Yeah.
I'm just curious whether there's any evidence for pollutants directly affecting odorant receptors or neuron behavior. I mean, I was almost kind of anticipating that you would talk about that, and you're talking about it changing the environment, which is fascinating.
So you have to get super high concentrations, and then it seems to be modifying kind of the receptor environment, but these are totally unrealistic concentrations. However, one thing that we have not talked about is that these nitrate radicals are creating smog, creating these particulates, and those actually can impact the respiratory system and perhaps the olfactory system of these animals. And this is interesting, these particulates are also modifying the plants. And so they're somehow getting into the stomata of the plants, and that in turn is producing an immune response in the plants and modifying their chemical communication process. There's a science paper on it, I think, recently.
And then we've got the last question down here. Sorry, one more.
Cool presentation. Do you have any hypothesis of the mechanisms that can help in the adaptation in have these olfactory responses in these polluted environments? So how moths can adapt to these new changing environments? Any idea, any hypothesis of how can they do that?
Yeah, that's a great question. One idea is that can be modified through the inhibitory, which glomeruli have the strength of these inhibitory connections. The other thing is that I'm really interested in going to Cairo or around there and seeing how, one idea is that some of these pollinators can start overexpressing these olfactory receptor proteins that are responding to ligands that are really differentially degraded. You know, so there might be some sort of regulation there, which would be really interesting, right? Super cool. Who knows?